This method can help us answer key questions in cell biology and biochemistry, such as how the dynamic assembly of protein-protein complexes controls signaling pathways and cell behavior.
The main advantage of this technique is that we can specifically isolate two interacting proteins while excluding uncomplexed individual proteins and competing binding partners.
Though we use this method to provide insight into receptor tyrosine kinase dimerization in breast cancer, it can also be applied to any other physiological setting or disease.
We first had the idea for this method when we realised the GFP nanobody recognised the three-dimensional epitope of GFP and other fluorescent proteins.
To begin, add 150 nanograms of entry vector, 150 nanograms of destination vector, and 8 microliters of TE buffer to a 0.2 milliliter tube. Next, add 2 microliters of recombinase enzyme mix, and centrifuge the tube briefly. Incubate the reaction for 1 hour at room temperature. Then, add 1 microliter of proteinase K solution, and incubate at 37 degrees Celsius for 10 minutes to stop the reaction. Next, thaw heat shock competent cells on ice, and transfer 50 microliters of the cells to a 14 milliliter round-bottomed polypropylene tube. Add 1 microliter of reaction product to the cells and mix gently. Incubate the cells for 20 minutes on ice. Then, heat shock the cells in a 42 degrees Celsius water bath for 45 seconds before returning the cells to the ice. Add 1 milliliter of LB media to the cells and incubate the